Welcome, everybody. My name is Gene Quinn. Thanks for joining us here today for this conversation about antibody genus claims, resurrecting that type of claim group and seeing what needs to be done, how we can do it. Is it possible, I suppose, even in the first place is the first question. And not surprisingly, I think we're going to say, yes, it is, but we're going to talk about how you can actually do that. This is obviously in response to several years ago, the Supreme Court, to some extent, my characterization took this industry out behind the woodshed and kind of tried to shoot it with Amgen. I just never understood that decision, but we have to live with that decision. And now strategies are developing so that we can live with that decision. So that's what we're going to talk about here today. We're not going to complain about the Supreme Court or Amgen. We're going to talk about what you can practically do in your practice today to try and address that new reality that we've been facing over the last couple of years. So with that brief introduction to the topic, let me kick it over to Renee to introduce our speakers. Renee.
Thanks, Gene. Today on our panel, we have Rich McCormick. He's the founder and principal of Innova IP Consulting. With more than 25 years of experience in pharmaceutical and biotech patent law, he previously served as a partner at Mayer Brown, where he represented major companies including Bristol Myers Squibb, Sanofi, and Regeneron in complex patent litigation and portfolio strategy. Today, he advises the life sciences industry on intellectual property issues related to biologics, antibodies, gene therapy, and other advanced immunotherapies. Next, we have Zach Silberscher. He is a partner at Silberscher Law and an experienced patent attorney with a practice spanning patent litigation, inter-parties review proceedings, prosecution, and licensing. He also frequently advises financial analysts on patent disputes in the pharmaceutical and high-tech sectors. Zach has been recognized in IAM Strategy 300, the world's leading IP strategists, and is regularly quoted in major media outlets on patent law and innovation issues. Next, we have Ben Doranz. He's president and CEO of Integral Molecular, a company focused on developing technologies that enable the discovery of therapeutics for difficult protein targets. He co-founded the company in 2001 and has led its growth while bringing multiple scientific technologies from research to market. Dr. Doranz is an inventor on 50 patents and the author of more than 100 scientific publications, with work appearing in leading journals such as Cell, Science, and Nature. And last but not least, we have Gene Quinn. He's a patent attorney and a longtime thought leader in the IP world. He launched IP Watchdog in 1999, which has grown into one of the most trusted voices in patent law and innovation policy. He is widely recognized for his expertise in software patentability and U.S. patent procedure, and he's been repeatedly honored as one of the top IP influencers and strategists in the global IP community. So you're in great hands with this panel. Back to you, Gene.
Great. Thanks a lot, Renee. And again, thank you all for being here for this conversation. It's an important conversation to have. I know for many of you, for some of you, it probably is an existential question about whether or not you can get the protection that you need. We'll have that conversation here today. If you have any questions during our time, please put them in the Q&A box. That's where I will be monitoring the Q&A box and try and get as many of your questions to the panelists as we have time for. If you want to chat amongst yourselves, and we encourage you to do that, please use the chat box. Sometimes the chat box gets rather active, you might say, and it is makes it a little hard for me and the speakers to follow along. So if you have something that you want addressed, please put it in the Q&A box. We do have Ayesh here who is going to be monitoring the chat and also monitoring the Q&A. So if you have a highly specific question, don't be afraid to ask it. Even if we're not going to be able to get it during the webinar, we'll try and get everybody questions to all answers to all of your questions, either online or offline after the webinar. So gentlemen, with that, let's talk about big picture to get us going. Ben, maybe I'll ask you first, give you the opportunity to lead us off. What is your big picture thoughts on this topic, antibody genus claims, or any aspect of that?
Yeah, thanks, Gene, and thanks to everyone for being here. We've been thinking about this topic a long time, and as a biotech company, we approach it from a data perspective. Can we discover, can we create the data that supports the kind of genus claims that our customers have been asking for? Actually, our own lawyers. As a company, we file our own patents on antibodies and have for 20 years. And our lawyers tell us, can you get us more data? Can you make mutants of this antibody? And we can. That's what we do as a company. And then as Amgen progressed through the courts and the Supreme Court and the decisions came out, we said, well, why don't we just give you all of the mutations of the antibody that we have? We don't have to just make a few. Here's all of them. And that kind of progressed into the genus claims that we're getting now. So we're really happy to say there is a pathway forward. It's, at least from our perspective, there's a lot of data that can be used to create better claim sets. And I think that's what I'd like people to take away, that there's a pathway forward through good data.
Great. Excellent. Thanks, Ben. And before I move on to the others for their preliminary thoughts, I want to just let you know, and many of you already seen it, I've posted an initial question to give us some idea of where your head is on this topic. So if you could just take a moment to do that, we'll go over it here momentarily. But Richard, let's go to you next. What are your preliminary thoughts on this matter?
Well, I mean, I've thought about this, and I don't want to be long or long-winded. There's a lot to say, but I think the future is, big data and AI. I mean, surprisingly, right? I mean, that's the answer to everything these days, big data and AI. I think that the CDR scanning that Ben's going to talk about goes a really long way to contributing to the pool of data that we can draw from. I tell my clients, look, don't swing for the bleachers anymore. You're not going to get a functional claim to an antibody without structure binding anywhere on an antigen, with some sort of clinical or physiological response. Those days are over. But I say, focus a little bit, right? Look at like these limited epitopic spaces. Maybe look at a single epitope that your lead candidate antibody binds to, and think about the structure that you need for a common structural features analysis in a way beyond just the amino acid sequence. So I think that what Ben's going to talk about, that technology goes a long way for us getting there. I think when you look at these enormous databases, the Oxford Protein Informatics Group maintains these huge antibody antigen structural databases. And that's the kind of data that AI can learn from to even further identify common structural features for antibodies. As long as we're talking about, your claim is focused on a very narrow epitopic space, along the lines of what we saw in Amgen, maybe even narrower.
Mm-hmm. Great. Thanks a lot, Richard. And Zach, your initial thoughts.
Thanks, Gene. So I was following the Amgen versus Sanofi case for several years, including attending many of the district court proceedings. And unlike unlike some people, I actually thought that the Supreme Court came out on the right side of this issue. And the big question that I think I'd like to talk about today and what we should be thinking about is whether or not this new era where we can no longer get broad functional genus claims, is that going to encourage biotech innovation? Is that going to discourage biotech investment? I think on balance, biotech innovation will actually increase in the new era without broad functional genus claims, in large part because it's going to reduce investment risk for companies that get sandbagged by broad functional genus claims because their competitor beat them to the patent office. I think this is actually a good thing for the industry. And what we're going to hear about today is a number of different options on how to pursue genus claims and no longer do what we did in the pre-Amgen era. And I think that the paratope mapping strategy that Ben in particular is going to talk about is never going to capture the same patent breadth as broad functional genus claims as the pre-Amgen era, but it provides a very interesting middle ground that is going to need to be developed and blessed by both the district courts and the federal circuit, but it's a strategy that's worth considering right now for all the reasons that we're going to hear about.
Great. Excellent. Let me share the results to our poll question here. And not a shock to me. Most people picked the largest number or major challenge followed by moderate challenge. Does this surprise any of you? And maybe also, how would you have answered this question? Richard, maybe we'll start with you this time.
Yeah, I mean, I think look, I think it's all depending on where you're sitting in terms of the level of technology. Amgen, the time of Amgen, major challenge, right? I mean, I think things have advanced exponentially since then. So what I'm going to be talking about is how maybe it's less of a challenge now. But I think if you're thinking about this 35,000 foot view, major challenge does not surprise me.
Yeah. Ben, thoughts?
Yeah, I mean, it's something again we think about from two perspectives. One, we can help solve these problems, but actually we're solving our problems for ourselves too, because
Hmm.
we patent and license antibodies. We do a lot of discovery. We don't do any clinical work, but we license them to companies like AstraZeneca and Context Therapeutics and others. And when we license those patents, the value is the patents. We're receiving significant licenses from major companies, and it's all based on the strength of those patents. So the stronger we can make those patents, the more valuable they are.
Yeah. And Zach.
I think it's definitely these results don't surprise me. I think it's definitely going to be a challenge to obtain broad defensible antibody claims, but we live in a new era right now, and I think that pursuing a panoply of strategies and to protect your antibodies, including outside of just claiming the antibody itself. I mean, what I've learned consulting with clients on how to delay biosimilar competition or generic competition is attack the problem in as many ways as possible with as many patents as possible. So we're never going to go back to the pre-Amgen era without some legislative change. But that doesn't mean that we can't protect our antibodies, even if the protection is more narrow.
Hmm. Great. Excellent. So I will stop sharing that question here now. And I think we're going to kick it over to Richard. We do have some slides prepared for you today to push our conversation forward. And Richard is going to lead off. So Richard, take it away.
Yes. All right. Hello, everybody. I'm happy to be here. My goal is just to sort of tee up the discussion that Ben and Zach are going to have. So I'm going to kind of just go over at a very high level the 112A landscape, post Amgen, post Juno, post AbbVie, and then talk about some patenting strategies, legacy, and maybe some old ones that have become new, means plus function, and see how those are going to fare against, in this world that we're living in. So, let me get down to my next page. Okay, so as many of you know, probably, last year was the 50th anniversary of the Kohler and Milstein paper publishing in Nature 1975 of the creation of the monoclonal hybridoma, without which I think maybe, we wouldn't be having this webinar. They stated in that paper with I think Nobel Prize worthy understatement that that the hybridomas could be valuable for medical use. And I think it turns out, they're absolutely right. I think that promise has been fulfilled. Paul Carter wrote a paper in Nature Reviews Immunology last year, kind of looking back at the last 50 years. So where we stand now, we have well over 200 therapeutic monoclonal antibodies approved by the various regulatory agencies across the world, tens of millions of patients treated. There's a global market value forecast about $500 billion by 2028 for the monoclonal therapeutic antibody market, and upwards of about 16,000 issued U.S. patents, have claims that are directed to monoclonal antibodies. So, so antibody therapeutics clearly on the rise. What about, the functional claims, which I think make up a lot of those 16,000 patents? I think what we have seen is sort of like a rise, decline, and fall. It's like the Roman Empire for functional genus claims to antibodies. I mean, I'll just do this very quickly. We start out with Regents. This kind of sets out the two different tests you can look at, which we know we'll talk about in a moment. Then we kind of move on to Noel v. Letterman. The antibody exception. If you have a fully characterized antigen, you can have a functional claim without having to satisfy the Regents test. AbbVie v. Janssen, sort of sub silentio, I think kind of abrogates that. They invalidate, IL-12 claims, functional claims, don't even give lip service to Noel. After that, we have Amgen v. Sanofi, the first Federal Circuit case in that litigation, where the Federal Circuit does abrogate the fully characterized antigen test. It's inconsistent with the statutory mandate for written description. After that, we have Juno v. Kite. You could see this devolution, right? Juno v. Kite is sort of like this further retrenchment of a rigid application of the Regents v. Lilly standard. And then obviously we end up with Amgen v. Sanofi at the Supreme Court for enablement, requiring full scope enablement. And now we've got these USPTO guidelines. I mean, they're basically saying we're implementing Amgen. We don't really have any further information for you about how to meet enablement, but we're looking at the WANS factors. So why don't we look at this? I'll do this again at a very high level. This is the Regions v. Lilly test, representative number of species, common structural features. And the thing that both of these prongs have in common is that you have to look at structure. So representative number of species, you need enough species, antibody species, say, to reflect the structural diversity of the claimed genus. Common structural features, you need a common structure that kind of runs through all of the species within the genus, so that when you see the claimed function, you immediately call to mind this common structure. And what I tell my clients, when I'm advising them about, for either of these tests, what the structure is, I say you have to think beyond, the primary structure, beyond the simple amino acid sequence. When I think about structure, and I think this kind of, where we are now with technology, I like to think about it as also tertiary structure, right? So the three-dimensional topography of the variable region, what that looks like. I think about, all of the non-covalent interactions that go on with antigen. So other structures could be, the way charges are distributed on the variable region, hydrophobicity mapping, or even just, spatial arrangement of polar R groups. So that's all structures we should be thinking about beyond just the simple amino acid sequence. And I think the goal overall that the Federal Circuit has stated for both of these tests is that they allow a skilled artisan to readily distinguish species that fall within the genus from those, and have the function from those that don't have the function, so that you can visualize or recognize the members of the genus. Excuse me. So here's the, sort of like the two leading cases applying the Regents test in the antibody space, AbbVie and Juno. I'll assume a certain familiarity with them. I think we could talk about these cases forever, especially Juno. But AbbVie is basically a functional antibody claim binding IL-12 anywhere on the antigen, has to neutralize IL-12, have a certain K-off rate. The species in that patent is they give you one parent antibody, Jo9, 200 variants of Jo9. They're all about 90% plus sequence identity to Jo9. And what the court finds, well, first of all, I should say, for common structural feature, AbbVie concedes there would be none. They put all their eggs in the representative number of species basket, and they say that the 200 variants are representative of the structural diversity of the genus. And the Federal Circuit kind of says, nope, not so fast. You've basically accused Stelara, which is Janssen's antibody, of infringing. Stelara has a totally different amino acid sequence. It's like 50% identity to the Jo9 and its variants. So Stelara is kind of like that, that Karl Popper black swan that falsifies AbbVie's argument that they're representative. You only need one example to show it's not representative, and that claim falls. We move on to Juno, which again is like this further retrenchment of a very rigid application of Regents v. Lilly. We could talk about this case forever. As Gene said, I'm not here to criticize it, but I would point out that at trial, the jury found that that Kite had not proven invalidity for written description. The district court denied JMAL on that and said, yeah, the jury could have found it met either RNS or CSF. And in fact, it could have even found that Europep exception, where this was a well-known genus, applied here. You didn't need to describe all the SCFVs because that genus was well-known. The Fed Circuit doesn't really buy that and validates the claim. We know it goes up to cert to the Supreme Court. They deny it around the same time that Amgen v. Sanofi is up for cert at the Supreme Court. At the end of the day, my feeling on this is that this is basically, if this claim was going to go down, it should have gone down on the point of novelty, which is whether or not you can activate T cells once a single chain variable region fragment binds to antigen. SCFVs were really well known. We knew how to clone variable regions by 2002 when the patent was filed. But anyway, I think that this is an example, of really like the true fall for me in written description for functional antibody claims. We move on to enablement, Amgen v. Sanofi, and the epitope claim. So I have this exemplary claim from the patent. And it's directed to an antibody, no structure mentioned, but it has a function, and the function is to bind two or more of these 15 amino acid residues on PCSK9. I've given you this very helpful surface model of PCSK9 where I basically have colorized the 15 residues, and you could see they kind of make like a very sensical epitope here. All the R groups are exposed on the surface. In terms of supporting this claim, they had 10 antibodies in their patent, 10 species supporting the claim. Most people note that there were 3 amino acids that did not have any antibodies bind to them, and none of the 10 antibodies bound more than 9 amino acids. For me, like when I was thinking about this case, and full disclosure, we put in, for our client when I was at Mayer Brown, our client Unified Patents put in an amicus brief coming down on the side of Sanofi. So that's just full disclosure. But when I was thinking about this case, thinking about when this patent was filed, which I think was like 2002, maybe more or less, was that when you do some quick combinatorial math here, 2 or more of the 15, it comes up to like 32,000 different permutations, right? 32 different thousand ways that, an antibody in theory could bind to PCSK9, at least within the scope of the claim. And, you've got 10, how many of those 32,000 are even make sense for an antibody being able to bind them? And I think there was testimony at trial that a lot of those were unlikely to have antibodies made to them. So that's how I always thought about this case. The Fed Circuit didn't really think about it in that way. But in any event, the important part for here is in the patent, the teaching, how do you identify undisclosed antibodies aside from these 10 that have the claimed function? And, Amgen set forth, these 2 methods, right? The roadmap or conservative substitution. The roadmap is basically pretty much do what we did. You just screen, and you'll find antibodies that bind 2 or more of these residues. Or conservative substitution, you start with an antibody that actually meets the function, and then you make, minor modifications to it. Maybe you're enhancing binding in some way, you're reducing immunogenicity. So that is the, that's how it goes up to the Supreme Court. And, we know how the Supreme Court came out on this, no enablement, a full scope enablement requirement for functional genus claims. And what the court said was that, Amgen's roadmap and conservative substitution methodologies, that's just basically giving the person of ordinary skill in the art 2 research assignments, essentially saying, do what the inventors did, go through that iterative trial and error process to find antibodies that bind 2 or more of those residues. That is painstaking experimentation. That's not the reasonable experimentation that the statute allows for enablement. But I think importantly, what the Supreme Court did say is that if the inventors had identified a quality common to every functional embodiment, a common structural feature, that would have reliably enabled, the skilled artisan to make and use the full scope of the genus. So I think the takeaway for me is that common structural features can satisfy both description and enablement. That's where we are now. Some of the claiming methodologies, maybe, after Amgen and Juno to think about. I think we still obviously have claims to, the actual amino acid sequence of the variable region or CDR sequences of your lead candidate antibody. Usually you claim those by seq ID numbers, and you could have functional limitations in there as well, but you have like a fixed structure. Claims like that obviously are the strongest on validity. When you're thinking about your LOE, you pretty much can assume it's not going to be before the expiration of that patent. And claims like this, as Zach mentioned, they're highly assertable against biosimilars. The problem, I think, is they're not going to capture a competitor's different antibody that may bind the same antigen, in or around even the same epitope, using the same mechanism of action for that downstream effect. So I think they're going to be of limited use in terms of replacing functional antibody genus claims. We also have these sequence identity claims, and we've all seen these. You claim 80, 85% to 99% sequence identity, compared to your lead candidate amino acid sequence, and you could have functional limitations in there as well. I think first, you've got, especially after Duke v. Sandoz, you've got some serious written description concerns. on risking invalidity for a claim like that, especially if you haven't figured out exactly like what does that field of 80 to 99% look like in terms of actual examples. Once you throw a functional limitation into a claim like this, I think you're also bringing in the Amgen analysis, and again, you're risking even more invalidity on enablement. At the end of the day, patent claim like this, it's broad, right? I mean, you may think it's going to get you what you want, and maybe it will approach a functional genus claim, but I don't think so. It's certainly easy to design around. If your claim is about, the 80 to 99% identity to a CDR or CDRs, you make a couple of changes to CDRs to design around, and, suddenly you're below 80%. But I think the real thing is that a claim like this is always highly, highly, highly unlikely to capture a competitor antibody. And I can tell you, I've been doing, aside from litigation, I've been doing FTOs and due diligence, for 25 years. I see patent claims like this all of the time. I've never come across a claim like this where I was able to match it up with a competitor antibody and say, that was a different antibody and say, oh yeah, this is within the 80%. It's never happened. So a claim like this is not going to be a substitute, for a functional genus claim. So that brings us to, this means plus function, which there's been a lot of discourse around. Is this an Amgen and Juno loophole or workaround? I won't go through the statute, but we all know the Ex parte Chamberlain case, which went up on appeal to the Fed Circuit in Re Zencor. The claim here was Claim 9, a method of treating a patient by administering an anti-C5 antibody comprising a means for, that's the language, binding human C5 protein. And what the appeals review panel did is that they basically said, yeah, this actually implements means plus function statute properly. It uses the right language. There's no structure in the claim. And the antibody species in the patent spec, this antibody 5G 1.1, that's the corresponding structure. And then what you're entitled to in terms of the scope of the claim is equivalence of 5G 1.1. Those would infringe the claim. And importantly, what the APR said was that you don't need to provide a written description for those equivalents in the specification. In other words, you don't need, even though it's a functional claim, you don't need to, there's no requirement to disclose a representative number of species or common structural feature for the equivalence to 5G 1.1. This was talked up. I think, everybody's talking about this case. Mark Lemley and Jacob Shurkow, mentioned this in their Yale Law Journal law review article, the antibody patent paradox, as a way maybe to get around, the jurisprudence on 112A. I have some questions about it. I think, first of all, this does not have the imprimatur of the Federal Circuit. The claim was ultimately rejected by the Fed Circuit, and the APR decision affirmed on other grounds. So the Fed Circuit never passed on this particular holding. So we don't know what the Fed Circuit would say about this, other than maybe to say that I think the APR did a pretty good job, shoring up the argument that you don't need to have description for the equivalence. Another question I have is whether or not you have to have enablement of the undisclosed equivalence. And I think there's some Fed Circuit case law that even Mark Lemley and Jacob Shurkow kind of acknowledge that suggests you still have to have enablement for that. And, how do you enable that, in view of Amgen? To me, the biggest question is even if your patent gets through, a validity challenge, it's an uncertain scope of equivalence, right? Like, is the equivalent of 5G 1.1, is that going to capture a completely different competitor antibody? Are we in DOE land? Is there some different type of equivalence here? And we just don't know. My feeling is that it's probably not going to be able to capture different competitor antibodies, but we don't really have any case law on that yet. But these are three sort of little asterisks I would put next to means plus function. So, the future of functional genus claims to antibodies, where are we? We've got the PTO guidelines, following WANs, following Amgen, but not really telling patentees how to get your claims enabled. But we do know that now common structural feature will satisfy enablement and written description. So the question at the end of the day is, here in 2025, how can we identify those common structural features? And I think we are, given big data and given AI, we're well beyond where the technology was when the Amgen patent was filed. when all of those patents were filed. I think, we're light years ahead of that now. So again, the first thing I tell my clients, and I'm just circle back to my big picture, is focus on narrower claims. Don't try to hit it out of the park. You're not going to get a general claim binding antigen anywhere. You might not even get a claim to the Amgen epitope scope, but maybe something that's a narrower epitopic space, or even like, a fixed epitope with like a single antibody. And I think that's where the CDR scanning that Ben's going to talk about comes in. And I think in terms of, how do you get those species? Clearly, like wet lab experiments, a methodical, comprehensive, systematic way of generating these antibodies in a high throughput fashion, and populating your specification with a lot of examples, especially the narrower the claim is. I think you're going to be able to find, at least from the amino acid sequence, a common structural feature, and Ben will talk about that. I think when you're thinking even more broadly, you look at big or bigger data, all these enormous databases we have, and applying AI to that and letting AI do what it does best, which is pattern recognition and finding common structural features across, a huge scope of antibody antigen interactions. So I will leave it at that and turn it over to Ben, or maybe to Gene for questions.
Um, no, I think we probably, looking at the clock, we probably need to move along to Ben. We'll get everybody answers the questions either here or offline, so keep them coming. But Ben, why don't you just take it away if you can?
Great. Thanks, Gene. Thanks, Rich, for the introduction and getting everything teed up. There's a lot of background here, and that covers a lot of ground. I just want to back up for a second to give a little bit of background about Integral, and this is relevant to the conversation. As a company, we've been around for 20 years in antibody discovery, engineering, and mapping. So we have a number of products and services all around the antibody space, a little bit in the virology space. A lot of people in the industry know us for our membrane proteome array, for specificity screening. But what I'm going to talk about today is actually around paratope mapping, which feeds into this into the IP discussion. And this started actually many years ago for us. We've been around for 20 years, and for much of that 20 years, we've actually been scientifically engaged in understanding how antibodies bind their targets, so epitope mapping and things like that. We've epitope mapped over 1,000 antibodies for customers and partners and ourselves. We've published many of those in over 200 papers. Actually, our customers and clients have also published those and patented them in many cases to get IP. So there's been over 200 papers and patents on on around the IP space, mostly on the epitope side. And actually, we published a paper a few years back in MABS, which talks specifically about the epitope mapping contribution to patent protection. At the same time, we've been doing antibody engineering for 20 years. So when the Amgen Sanofi case came out, and during the evolution of that case, we realized that there was a fundamental shift in the landscape for patenting antibodies. And we also realized that we may have some of the tools that can answer the questions that were left by the court cases. In particular, as everyone knows, the court kind of struck down functional claims and said you can't do this anymore. No longer stands. You need a common structure. You need reasonable experiments, et cetera. But they didn't provide a pathway forward for it. And so we published this paper that resulted in us thinking about this question and publishing this paper in Nature Biotech about a year ago, describing the CDR scanning for intellectual property protection around antibodies. And again, the paper, I think there's a link in the chat if anyone wants to download it. But the problem obviously was that, you want to claim more than one species. You can claim your antibody, the exact sequence of your antibody, but that's pretty easy to get around, and it's not really claiming anything around the genus. You can't claim a genus anymore, at least as defined pre-Amgen, that binds claiming every antibody that binds that target. That's just that's not reality in the future, as Zach has kind of pointed out. The Amgen case kind of changed that landscape. So you can't do that. So our challenge became, how do you claim a genus that meets the USPTO guidance for enablement and written description, but do it in a way that's reasonable, that meets the premise of the law, the quid pro quo, the guidance of the courts. And the paper describes a strategy, more theoretical at the time, and I'll show you the reality at this point, where we could take an antibody and experimentally in the lab define an antibody's genus and its variants using a common structural feature, which is basically the paratope. So this general strategy is borrowed from what's been around for a long time around chemicals. Generally, it's well documented, and many people have patents on deriving patents around small molecule scaffolds. So you've got the scaffolds, you can put R groups everywhere. There's limitations around those R groups, but you've got the scaffold and the acceptable substitutions around that scaffold. And what we realized is that for antibodies, which is what we specialize in, the equivalent of a scaffold is called the paratope. It's the critical amino acids of the antibody that you can't change. That's the common structural feature, this paratope. And they're in red here. They're the residues that bind the epitope generally. So if you start with the premise that the paratope is the common structural feature, and that's what the courts want, we can define what the paratope is. Like that's experimentally feasible. So that's the premise. If we can take an antibody, we can mutate all of the CDR residues to every other possible residue. And of course, for antibodies, there's only 20 amino acids per residue. There's only 20 substitutions versus like a small molecule where an R group can be anything. It's an infinite number theoretically. But for antibodies, you've got 20 residues you can substitute at roughly 50 different positions in the CDRs. You can make those variants, and you can define in blue here what's permissive and also what's not permissive. What can't you change? And that gets you to an antibody genus. So in these cases, for instance, you can change this arginine to a cysteine, a glycine, a histidine, but you can't change this proline. That's one of the common structural features. And that translates directly to patent claims. So if you kind of analyze this, it gives you enablement and written description. Like there's the data, you've got the antibodies, you've got the variants, and that translates directly to the claims. So that was all in theory. And then in practice, we went into the lab, and that's what we did. We took an antibody. It's against a particular G protein coupled receptor that we work on. We have a lot of robotics in the lab that do a lot of these high throughput screens for us. But we essentially did that. This is a portion of the data, mostly the heavy chain, where we took every parental residue of the heavy chain. We changed it to every possible substitution. And we could see which residues you could substitute, like in blue. These are perfectly acceptable substitutions, but also which residues you can't substitute. Like take this 155 here, you can't change it to anything else. It has to be a glycine. So that's the common structural feature, and you can kind of go down the list and see how that works. What's also really interesting from both a scientific perspective as well as an IP perspective is that you also end up with novel IP because you end up identifying mutations in orange here that bind better than the wild type. And that's exciting from a novelty perspective. But in any case, we took that data, we took those proof of concepts, and we ended up launching a service where you can mutate the antibody, you can submit sequences, we mutate the antibody, every variant, we express them, we quantify them by ELISA in the lab, by expression and by binding. It's all normalized to expression. And that ends up in a report describing all that data, the paratope, the common structural feature, the permissive substitutions, enhanced substitutions with improved binding. And we even actually write the patent claims for you in that report, because it turns out when we started working with clients, it's a lot of data processing, and rather than them sifting through giant Excel sheets, we've automated it so you can push a button and turns that data into claim sets immediately. So it takes about 12 weeks. We generate about 1,000 variants. And if this is, by the way, not a new concept, mutating antibodies and deriving lots of variants is not a new concept. But the new thing here is that if you did this, even 5-10 years ago, this kind of project of 1,000 variants across an antibody would probably cost you about $500,000 to $1 million. And it's been done by major pharmaceutical companies and published. The novelty that we've introduced is that we can do that for 1/10 of the price. So we can do this. Our typical projects are $50,000 to $100,000, and it gives you all this data and all the IP around that antibody. So we launched this service, and the immediate question we got, especially from lawyers, is, well, can you get a patent out of this? And the answer was yes. We actually just got this issued about a month ago. So we filed this patent based on that initial antibody. There's a link here, and we'll send out that link with the follow-ups here, but you can see the patent, the annotations, the claims, the USPTO office action, and all the file wrapper information if you're interested. But I'll just summarize it here. We got our genus claims allowed, and they basically cover the permissible substitutions as well as the theoretical combinations of those substitutions, which I think is really important. And we'll talk about these just briefly for a minute. So the first set of these permissible substitutions. So this is the actual data we generated. I'm just taking a portion of the data and the claims, of course, just to highlight the principle here. But we said, you can substitute this serine for all these types of amino acids, but you can't substitute, for instance, this glycine with anything. So that's the data. We've reduced it to practice. It's a common structure because we've identified mutations, residues, positions that you can't substitute. And it's all based on structure, not function. And this is the claim that we got allowed. Again, you can look at it directly, but this is the sequence. There it is. A variant thereof. This one just taking a piece of the claim, of course. And you can change that particular, in this case, the first residue, serine, to any of these other residues. And that's the claim we got. And that's very consistent with the Supreme Court and USPTO guidance at this point, where you have to provide data and a claim set and support that enables a person skilled in the art to make and use all of what's claimed. So we're claiming these substitutions. There they are. There's no real controversy around enablement and written description for all these variants. We have the data. They're all in the patent. The second part of what we got in the patent is same claims, same data, but it's the combinations of those substitutions. So in this case, we got not just that you can change this residue to all these other residues, but you can change all of these residues to different combinations in combination, except for the common structural feature residues like this glycine. There are no substitutions for that glycine. So that's the common structural feature in our mind across all the CDRs that the Supreme Court, the USPTO is looking for. This allows you to visualize, recognize the members of the genus. They have to have this glycine here in this case. So there's the general quality running through the class. And that's why we got this claim. This is allowed, comprising the amino acid sequence of this selected from these residues and or these other residues. So the other thing that came out, the other question that we sometimes get for this is, well, you're predicting these theoretical combinations, but is this reasonable? Is these common structural features, are they actually allowing a person skilled in the art to build the genus? And is that going to allow a person skilled in the art to do it in a reasonable way? And we didn't know that at first. I mean, theoretically, you should be able to build the genus around these common structural features, but we didn't know until we tried. But when we did try, that's exactly what happened. So this is an example using that same antibody where we took those common structural features, we held them constant, but we built about 400 other mutations, changing the other residues that were permissible around the common structural feature. And 95% of those variants are not only as good as wild type, but they're as good or better than wild type. As a matter of fact, in many cases, we identified variants that are 30 times better than the parental, so new IP at the same time. So we think this answers the question that the courts and the patent office have give guidance to, which is you don't need to make every variant, every single embodiment within the claims, within the class, but you need to identify the common structural features that enable you to make and recognize the genus within a reasonable amount of experimentation. And any person looking at this would say, yeah, if you can predict the genus 95% of the time, even 50% of the time, that's pretty reasonable in most courts' eyes, I would think. So backing up a step, and I know Zach will take over and talk more about this, but when we look at our strategy, we think it's a nice balance of what the court systems that are intending, which is that quid pro quo where you've got claim breadth, maybe not everything under the universe, but around your antibody and anything around those common structural features. And you've also got S112 compliance compared to the other techniques or approaches, strategies that are out there. And this has held up really well. We launched this service last year. It's all fee for service. And we had really good engagement and really good feedback. You know, our job is to make everyone happy, and providing this data. And what we found so far is that we're at least making some of the examiners happy. We're checking off the boxes for enablement and written description. The management, the CEOs we talk to, they understand the value. They're getting stronger patent protection, new IP. The investors really like it because actually some of our customers are VC firms that are building better patent portfolios for their companies and reducing the FTO risks. And the scientists really, really love it because kind of a whole nother topic, but they get better information for antibody engineering, efficacy, safety, and developability. So far, this has been really well received, and we're excited to work with people. Thanks, and thanks to everyone who's contributed to this, and I'll kick it back to you, Gene.
Great. Thanks a lot. There's one question that you might just want to ask that probably have an answer that some people may have in their minds. The question is, so what separates the projects that you file on versus the ones that your clients file on?
There's only a few targets that we go after as a company. So if we recognize that there's a target that someone comes to us that we can't go after, we flag that immediately and don't do the project. And we have some, for instance, we have some licenses, AstraZeneca, Context, and Conjugate, and a few other companies where we've licensed our IP to those companies, and the licenses prohibit us from working on those targets. So if someone brings us that target, actually, if they even brought us our antibody and said, "Can you do this fee for service?" You know, we would flag that and say, "Well."
So it's just it's a carefully choreographed
narrow
conflict checking type process. You're not in competition with the people who are offering fee for service.
No, we've been in business for 20 years, and we're pretty conscious not to be in competition with our customers.
I figured that was the answer, but the question was asked, and I think it was something that probably just nailed down. But thanks, Ben. Zach, let's kick it over to you so you can go through your slides and presentation.
Sure. Okay, well, thank you. I only have a few minutes left, and so I want to cover some of the most important points of my presentation. I think that Richard did a great job of providing a background on how we got to, of the law on how we got to Amgen Sanofi, as well as identifying patenting strategies for pursuing genus claims in the wake of Amgen Sanofi. One of the most important of which is the strategy that Ben talked about, including paratope mapping. There's two questions I'd like to address. That part of where I'm coming from is a lot of my clients are investors in the pharmaceutical industry, including analysts that talk to companies on both the buy side and the sell side. And the question is, first of all, whether or not the fact that we can no longer do broad functional genus claims, is that going to discourage biotech innovation? And then the second question is, given the array of current strategies, like the ones that Richard discussed, like which ones are going to encourage the most biotech investment and which ones are going to move the industry forward? So really quickly, with respect to biotech innovation, like I mentioned earlier, I do believe that the Amgen Sanofi decision actually will encourage more innovation. What was interesting was that during the Supreme Court case, while it was pending, there was a number of amicus briefs filed on behalf of both sides. Interestingly, the amicus briefs filed on behalf of Amgen, mostly big pharma companies, argued that without the ability to file broad functional genus claims that could basically patent an entire class of new inhibitors, that's going to kill the incentives to do research and development into life-saving therapies. As expected, the amicus briefs filed on behalf of Regeneron and Sanofi argued the opposite. And I think what was most interesting takeaway was that broad functional genus claims, even though they might have seemed to be a boon for the companies that held those patents, created major investment risk for for their competitors. And Regeneron was an example of that. I mean, Regeneron faced an injunction against its PCSK9 drug after investing hundreds of millions of dollars, not because it had inferior efficacy or toxicity issues, but really because Amgen beat it to the patent office with these broad functional genus claims. And in fact, when I was at the district court hearing on the injunction proceeding, the judge actually made a remark about that. She said, I don't really see that Regeneron did anything wrong here. I just see that Amgen made it to the patent office first. And that kind of having a simple one patent potentially sink $100 million investment spooks a lot of investors in any industry. In fact, when you look, one of the interesting things that comes out of the Amgen versus Regeneron and Sanofi case is that even though Amgen was not able to enforce its broad genus patents against Regeneron, even though it lost those patents at the Supreme Court and it could never enjoin Regeneron's Praluent drug, Amgen's business case for Repatha was not undermined. It's still actually there's some data that Amgen is actually beating Regeneron in the market today. And I think that this is important as we go forward and realize that even though we can't get these enormous breadth claims that we had before Amgen, that doesn't necessarily mean it's going to kill the business case for our drugs and our pharmaceuticals going forward. And I think that's a point that Richard was making. We don't have to hit it out of the park anymore. Get narrower claims that are just as useful and use them in a smart way. Richard talked a lot about the cases leading up to Amgen. I thought I'd identify a couple of cases in the wake of Amgen that sort of illustrate that this isn't really going anywhere, and the way that the courts are thinking about the post-Amgen environment. The Buxalta case is really just an instance of the Federal Circuit applying Amgen's analysis against broad functional genus claims, much like the ones that were at issue in the Amgen case. Richard also mentioned the Duke versus Sandoz case. What's interesting about this is that it was applying the Amgen reasoning within the context of a small molecule patent. So you can see some read-through outside of the antibody scenario to sort of narrowing the requirements for both written description and enablement. Another interesting case that is still pending that I've written about is a series of post-grant review proceedings that were filed by Merck against patents that might soon be asserted against Merck's oral formulation for its cancer drug, Keytruda. What's interesting about these PGRs, which have now been instituted, is that they included patents involving the sequence identity claims that Richard discussed. And even though the structure of those claims required 95% identity to reference proteins, Merck made the point in those proceedings that the claims still encompassed billions and billions of antibodies. In fact, they argued that if you were to actually manufacture every single antibody that was encompassed in one of these claims, the aggregate weight of all those antibodies would be more than the planet Earth, which was their way of illustrating the magnitude of antibodies that could be included even in sequence identity claims, which is currently being entertained as like an alternative to pursuing genus claims, given that we can no longer do it the pre-Amgen way. Another interesting point about this case is that the patent owner tried to circumvent the written description and enablement analysis by arguing that the claims themselves are only directed to structure. There's no function. You don't have to prove any activity. And if you don't have to prove any activity, you don't have to test them. That I think Richard hinted at this, which is whether or not, the claims have to have a functionality or not. But without a functionality, you're going to probably run into a utility problem. The argument was rejected by the PTAB under a claim construction analysis, but it's probably going to go up to the Federal Circuit at some point. It's probably not a winning strategy. So briefly, Richard and Ben talked about a series of new patenting strategies in the wake of Amgen. And I think that it's important to sort of look at them all at once and we see that they each have a trade-off between the breadth of patent coverage on the one hand and 112 issues on the other hand. Obviously, functional claims like those in the Amgen case are no longer viable, barring a legislative change. Parental sequence claims, I think it's prudent to always get claims on that for the sake of biosimilars and just to have them in general. There's no reason not to, but they're not going to give you the broad coverage you need in a genus way. The sequence identity claims, I think, as illustrated by the Merck case, present a lot of the same written description and enablement problems, given that they might encompass so many antibodies. Means plus function, I think one point I wanted to make about that is that although they seem to have broad coverage, because they include structural equivalents, those structural equivalents a lot of times have to be known and identified before the patent issues, which means that if you were to assert them in litigation and your competitor's antibody might actually have been prior art to your own patent. That's why I think that integral molecular's approach provides this interesting middle ground between patent coverage on the order of thousands of potential antibodies as opposed to just a handful of antibodies. And because it identifies that common structural feature that Ben talked about, namely showing what is essential in the paratope, that could potentially satisfy the written description requirement and the enablement requirement. Obviously, there's a lot of open questions. I think the one open question that Ben identified is that they got claims on combinations, even though they don't necessarily have the data. Is that going to satisfy the enablement or written description? I think that that's an open question that's going to have to be decided by the courts. But one thing I've sort of learned by working with Ben is that the technology is developing to include those combinations. And so what's interesting about this space is that the technology might be moving faster than the court decisions. And so as we move forward, we might not get certainty from the Federal Circuit for another 10 years, but the technology is catching up so quickly that I think that it's important to stay on top of it. And I know I only have 1 minute left. Big picture, I think it's important to pursue as many strategies as possible, given that we're in this landscape now without any blessing from the Federal Circuit on anything. So I'm gonna stop there, given that we only have 2 minutes left.
Okay, great. We may go just a little bit long. There's a couple of last remaining things that I want to make sure that we get to. There is one question, Ben, from Sarah about, do you have any thoughts on expanding from proteins to nucleotide acids or nucleic acids?
Yeah, our focus has always been antibodies, but the approach and the strategy we're talking about actually is beyond antibodies. You could do any protein and anything encoded by a nucleic acid. You can start making substitutions and get the same kind of protection. And actually, in the Nature Biotech article, we kind of alluded to that. You can do viruses, you can do genes, RNA. There's a lot of other modalities these days. We focus on antibodies, but the same strategy applies.
Okay, so if anybody has any questions beyond just the antibodies, they can still get in touch. Okay. And then what I would like to do, and I know we're just 1 minute out, but I want to give everybody an opportunity to wrap up because I think the presentation was excellent, knowing that we're not going to get to a lot of the questions. We will, as promised, get all of you answers as we end and move into the post-webinar phase of getting you the materials and the recording and the PowerPoints and all that sort of thing. So we'll get everybody questions answered, but I'd like to give each of you an opportunity to wrap up as I launch the final poll question here. So Ben, we'll give you the final word. Zach, how about we start with you? What is your takeaway message today?
I think the big picture is that we're kind of in a new era. We can no longer get these broad functional genus claims that we had before, but that doesn't mean that it's going to kill the industry, and it doesn't mean that we can't get strong patents that are going to protect our IP, not only against biosimilars, but also against competitors. And I think that the paratope mapping strategy and the technology that's developing is happening so quickly that it's worth pursuing. And you really have to pursue all strategies. And because at the end of the day, we don't really know where the federal circuit's going to come out in 10 years.
Right. And that's not just this area, that's across the table. I mean, it's sort of like all of the above patent strategy because there's not a lot of certainty. So thanks a lot, Zach. That was great. Appreciate you being here. Richard, your message. If there's one thing people will leave today remembering, what do you want that to be?
I think I just want to, I mean, I agree with what Zach just said. I want to add, maybe a little extra value to the CDR scanning and the, and the product that you're going to get from Integral Molecular. You're going to get sequences, right? And amino acid sequence is a structure, and that's a common structural feature. I think we're at the point now where you have AI programs like AlphaFold and even better that can take those sequences, and you could even expand the common structural feature beyond the primary amino acid sequence. You can plug those sequences into AlphaFold. You can get really, really good results that probably are almost as good now, if not as good as X-ray crystallography, electron microscopy. You can see the three-dimensional structure, and then you could work off of that to look at like charge distribution, et cetera. And I think you could even have broader claims, to different kinds of common structures beyond just the amino acid sequence. So I think I see that as like an added value to what Integral Molecular is providing you as a product.
Mm. Excellent. Great. Thanks a lot, Rich. And Ben, the final word goes to you.
Yeah, thanks. First of all, I just want to say thanks to everyone, Gene, Zach, and Rich. By the way, I appreciate the endorsements, Zach and Rich. And by the way, they're not being paid to say that. We do share common views on these things, but I appreciate being part of the webinar. I think what they said is exactly right. The past is the past. It's not coming back. The future to us looks like data. And I think any scientist, any, IP attorney that works in this space knows like data drives not just good results and clinical products, but also good IP. And that's the direction the courts are telling everyone to go. As a company, we're kind of agnostic to what the courts and patent office and Supreme Court say. That's just the rules we're following. But within the rules, the guidance they're giving us, that's the direction they're pointing from everything we're telling. And it's exactly what's been done more or less for small molecules. We're just applying it in a different field. So if we can help you, great. But it seems like, whichever way you go, there's kind of limited strategies going forward. And this is relying on data is never a bad strategy. So we hope we can help people.
Certainly in this area, relying on data is an absolute necessity. So thanks a lot, Ben. Really appreciate you being here. And I appreciate everybody in the audience being here and sticking with us a few minutes to give us an opportunity to wrap up our thoughts. I thought this was fascinating. If you asked a question, we'll get you an answer. But that's all we have for today, everyone. So bye for now, and thanks for coming. We'll see you soon.
All right.